There we go. All right, so today, I'm trying to think where we are. So you guys have a quiz today on chapters, 8, 9, and 6, right? So micro, so 8 would be all of metabolism, 9, DNA replication, transcription, translation, and then 6 is viruses, if I'm not mistaken. A reminder that I put it in the announcement, but just to remind you again, for chapter 9, you will need the mRNA codon chart. I would recommend that you have one that is a physical chart so that you can have it next to you, because as a reminder, every time you leave the screen to go do something else when you're taking a test, it records that as that you're leaving the screen. So students who then complain that they don't have enough time to complete the test, if I look and I see that you spend a lot of time not actually on the test, but doing something else, then obviously that's not going to make me very sympathetic for the reasons that you can't finish the test. So that aside, I would get a copy of the messenger RNA chart and I would have it right next to you so that you can quickly answer those questions that are going to involve using the chart. I think there's maybe 2. It kind of depends on which questions you get. And there's, so you should have a chart with you. That's important. And the other thing, I think the lab safety is open. So you guys can do the lab safety and the lab safety quiz, and then you can start playing with your little adorable microscopes. The first exercise is basically just to draw pictures of what you see from the included slides, which looks like it's onion, wool, and frog blood. So I'm going to ask that you do that and then make one sample of your own. So you can do a hair, you can do cheek cells on the inside of your mouth with a toothpick, and you can stain them with crystal violet from your stain set. I would advise, if you're going to, when you do that, to put a drop of water on the slide first before your drop of crystal violet, because the crystal violet's pretty concentrated. And if you just put the crystal violet on there, it'll be too dark. You won't be able to see anything. You should be able to see your cheek cells. And then you should also be able to see little tiny, they'll probably look like just little dashes. Those will be the bacteria that are in your mouth. So it works best if you do it before you brush your teeth or after you've eaten something and not brushed your teeth, but that aside. Okay, so questions that you have for the pending doom, because if you don't, so let's start with that. So questions you have for me. Yes, no, maybe so. If not, I have some questions for you that you can think about that may or may not help you on the test. But I was thinking about it this morning while I was exercising, and I was thinking, well, if no one asks me any questions, then I'm going to start asking questions of them. So I'm going to just, oh, sorry, go ahead. I can't hear you, whoever that is. Kanika, I can't hear you. Can you turn up your volume?
Sure.
There we go. Gotcha. Did you have a question particularly?
No, I'm sorry, I didn't. I think I just jumped on late. I apologize.
That's all right. That's all right. Then
sorry.
Hit your microphone back off. I'm going to ask you guys some questions.
All right. Thank you.
And I don't know everything on your screen. Okay. Yeah. Well, I want to share a whiteboard. There we go. So I'll just share this with you guys. Can you guys can see this? Yes. Somebody just let me know.
Yes.
Okay, great. So I'm just going to bring this down. And so for chapter 8, there are a couple of things that I would mention in particular. And again, if I'm asking you questions, these are going to be questions that are probably a little more complex than what is going to be on the test, but the idea is to help you with some of those higher order thinking questions. Okay. So one of my favorite questions, and I always ask this in Bio 1 as well, like usually the day before the test, same as we're doing now, is I like to ask students, if you had a fat, so a lipid, right? And that lipid, remember, fats are 3 glycerol and 3 chains of fatty acid. Those fatty acids can be broken off in groups of 2, which, so there's usually 3 fatty acid chains, unless it's a phospholipid, right? Then there'd be 2, but I digress. So there'd be 3 fatty acid chains. And remember, those fatty acid chains are hydrocarbons. So it's carbon like this with hydrogen, carbon with hydrogen, carbon with hydrogen, right? All the way to the end. And there's usually numerous of them. Let's say, 12 in a chain. And then in the other next fatty acid chain, there might be 16. And in the other one, there might be, whatever, 12 again. What I want you to think about is you can break those fatty acid chains, or not you, your cells or bacterial cells can break those fatty acid chains into sets of 2 carbon, and they can be converted into acetyl coenzyme A groups. This is why fat stores so much more energy than sugars, because even for each glucose that you can break apart, you're only going to get, remember, 2 acetyl-CoA groups from each glucose. So even if you have a polysaccharide, right, like starch or cellulose, right, or glycogen, even if you have a polysaccharide, and remember, those are made up of repeating glucose units, right? And let's say there's even 50 glucose units in there. Each glucose is only going to give you 2 acetyl-CoAs for the Krebs cycle, right, after the prep step. When you break down this fat, the example that I even just gave you, right, I think I said 12 hydrocarbons and 16 and 12, right? So if we add that up, I've got 38 acetyl-CoAs from a single fat. And that's not counting the glycerol, which will also get broken into an acetyl-CoA. That one third carbon will get taken off of there. So you can see pretty quickly, right? Even if I have, maybe this wasn't a good example. Maybe I should have used fewer glucose in my starch. Let's use 30 glucose in my starch, right? If I have 30 glucose units, that's still only going to give me 60 acetyl-CoAs total, and 1 fat with just 12, 16, and 12 fatty acid chains already gives me 38, or more than half of that, right? So the point I'm trying to make is that fats store a lot more energy than even complex polysaccharides. Now, given this, I'm going to erase this polysaccharide business because I don't want you to look at that or be confused about it. Given this, let's stick with this 38, these 38 acetyl coenzyme A molecules that can be produced. Well, actually, it would only be half of that, right? So this is 12 carbons, 16 and 12. So let's say 38 carbons in the chain. And then that would, 38 divided by 2 is what, 19? Right? Yeah. So that would be 19 acetyl coenzyme A molecules. So if I had a lipid that I could break down into 19 acetyl CoA molecules, the question that I would pose to you then is how many ATP total, so I would do that one last, how many NADH are produced and how many FADH2, and we can even go with how many carbon dioxides are produced from this fat. And let's not worry about the glycerol being broken down into another acetyl-CoA. Let's just stick with the 19. Okay, how many of all of these things, if these 19 acetyl-CoA molecules completed the Krebs cycle, TCA cycle, citric acid cycle, and of course the electron transport chain? So I'm going to let you stew about that for a minute. See if I can time myself and not talk for a minute, which would be tough. Let's see if I can just get rid of some of this so that I have more space to write. So where should I start? I'll just give you some advice, right? So what I would do is the chart that is, I don't know if that's in a handout that I gave you or not, but the chart that I constructed probably during one of the lectures that included glycolysis, the prep step, Krebs, TCA, right, citric. That chart that listed how many ATP, how many carbon dioxide, how many NADH, how many FADH2. And I think I probably suggested that you add columns to that chart about the starting molecule and the ending molecule for each step, and also where in the cell for eukaryotes and prokaryotes, right? So we know, just sort of as a refresher, we just go through glycolysis, we net 2 ATP, right? The prep step, we don't make any ATP directly, the Krebs cycle, and this would be if we're starting with glucose. So this is a little tricky because we've got 19 acetyl-CoA molecules, remember, that we're trying to follow. But if you had this chart in front of you and you could remember that if I start with glucose, I have to go around the Krebs cycle twice. So that's going to give me 2 GTPs that are going to be converted to ATPs in the Krebs cycle. For glycolysis, I believe it's 0. The prep step, we release 2 carbon dioxides, and for the the prep step is 2. Krebs cycle, I believe we do 4. Which is a total of 6, because remember, glucose is C6H12O6, so that gets rid of the carbons and the oxygens for us. NADH from glycolysis, I believe it's 2 from the prep step, and then 6 going around twice. So that's going to give us a total of 10 NADH that are going to go per glucose to the electron transport chain to produce those 30 ATP. Plus we have, let's use the yellow, plus we have these 4, right, in the happy yellow suns, happy yellow sun. And then remember our FADH2 molecules, we've got 0, 0, and we produce 2 when we go around twice. Those also go to the electron transport chain, but they produce fewer ATP because they join the electron transport chain later. So they're only going to give us, rather than 3 each, that's how we got from the 10 to the 30. Each NADH gives you 3 ATP, right? Each FADH2 only gives you 2 ATP, so that's going to give us an additional 4. That's how we get to our 38 glucose per, or 38 ATP per glucose. Okay, so why did I go through all this other than to remind you of this? And that you need to have this chart for chapter 8. You need to have that like memorized like immediately so that you can use it to answer a question like this if a question like this shows up on the test, right? So erase that. So now if I have 19 acetyl-CoA molecules, the question becomes, where do I start and how do I use this chart, right? So if I have acetyl-CoA molecules, then clearly I'm not going to start with glycolysis. So I'm not going to produce any of these molecules, right? Acetyl-CoA is at the end of the prep step. So I'm not going to produce any of these molecules either. Acetyl-CoA is ready to go right into the Krebs cycle. So put an arrow like that, because it's going to be going right into, that's the starting molecule, if you want to think of it that way, for the Krebs cycle, or the citric acid cycle or the tricarboxylic acid cycle. And if I do one turn of the Krebs cycle, how many ATP, NADH, FADH2, and CO2 do I produce? Well, we know this is 2 turns because this chart is based on glucose. So we can very easily say, okay, we would make 1 ATP and we would produce 3 NADH for 1 turn of the Krebs cycle. So let's put that here. How many for 1 turn of the Krebs? Right? 1, 3, and we're going to make 1 FADH2, and we're going to release 2 carbon dioxides. I don't think I have any other colors. Here I have red. 2 carbon dioxides. Okay, so if I go around the Krebs cycle 1 time, I get these many things. I'm going to go around 19 times because my lipid that I've digested, right? My lipid has 19 acetyl-CoA units. So first thing I'm going to need to do is multiply each of these by 19. All right, so 1 times 19, that's easy. So that's 19 straight up ATP. 3 times 19. Dr. Stewart is not a mathematician. 9 times 3 is what, 27, carry the 2. 3 times 1 is 3 plus 2, so that's 57. NADH. Whoops, let's make it the right color to help you. Keep it green. 3, what did I say, 57 NADH. I'll have 38 FADH2s. No, I won't. Just 1, right? 1 times each. 1 times 19, that gives me 19 FADH2s. And then I will have 2 carbon dioxides. Okay. So 2 times 19, that's the 38 that I was looking for. All right, so that means this one's finished. 38 CO2. If I turn this all the way to completion, meaning if all of these, so this would be literally this would be the first part. So I would have 57 of these of NADHs, 19 of FADH2s, and 19 directly ATPs. Then if I took the NADHs and the FADH2s and sent them all the way through the electron transport chain. All right, so instead of 10 up there at the top, we're going to put in our 57. And for every NADH, we get 3 ATP. So 57 times 3 times 3 is 21, carry the 2, 15, 16, 17. So now I've got 171 ATP from the NADHs. And from the FADH2s, remember I get 2 for each FADH2. So that would give me 19 times 2. So that would give me 38 more ATP. So if I completely oxidize this lipid, right, it's actually called, I think, beta oxidation, just like the complete oxidation of glucose is cellular respiration. If I completely oxidize this lipid all the way and produce the most ATP that I can from it, I'm sure you can do this faster than I can, right? 171 from the NADH, the 38 from the FADH2s, the 19 that I get just from going around the Krebs cycle with that substrate level phosphorylation. This is going to be my total number of ATP now. I've got 18, carry the 1, 10, 11, 12, carry the 1, 228 ATP from oxidizing that lipid. This might now be a better time to compare to my carbohydrate example where I had, well, let's see, if I have, never mind, let's leave it at that. Would I ask you to do that whole thing on a test in 45 seconds or 50 seconds? I think I've increased the time on this one to 50 because there are some things that you're going to have to actually use that messenger RNA chart for. It's going to take you a couple more seconds. No, I wouldn't ask you this entire thing as a single question, but I might ask you a piece of it, right? I might ask you I might ask you this portion, right? How many times or how many raw materials would you get if you had a acetyl-CoA molecule that had 19, and if you had a molecule that you digested and it had 19 acetyl-CoAs? Or I might say, if a molecule, lipid that was oxidized produced 57 NADH, and those NADH went around the Krebs cycle, how many ATP would be able to be produced from that? You should be able to do that math pretty quickly. Remembering, of course, that every NADH is going to give you approximately 3 ATP. All right. So yes, this is a question that's much more difficult than what I would ask you on the test. But if you can go through a couple questions like this, that will help you be ready for all of these kinds of things, right? If you can reproduce this chart very quickly and remember adding on the starting molecules and the ending molecules, as well as where it happens in the cell, that will help you a lot to be able to answer any of the higher order questions that are a little more complicated than something like, how many ATP do you net from glucose, right? That's like a knowledge question now. That should just be basic. You should just know that off the top of your head. Questions, comments, worries, concerns before I move on? Yes, no, maybe so.
I have a question.
Yes.
No. Okay.
No,
Well,
go ahead. I can put it back.
I'm not sure if there's like an easier way to, so is it just kind of making sure that we know how many enzymes or how many ATP goes around in each cycle? Or should we just memorize this whole thing? Like, is there an easier way to
No, there's not
navigate?
an easier way.
Okay.
No. So the lecture that goes with this, I would have gone through each of the parts, each of the steps, and
Okay.
gone through the starting molecules, the ending molecules, and where these ATP are produced. You are not responsible for the names of the enzymes or how many enzymes are in each
Oh.
step.
Okay.
There's a list, again, in that video for chapter 8, there's a list of what you needed to know from this particular portion, from the complete oxidation of glucose. There are other things in chapter 8. There is also a whole section about enzymes.
Right.
And there's also a section about integration of metabolism that would include the fermentation, the anaerobic respiration, and then something like similar to this that I'm talking about now, this sort of integration of how lipids get used for energy or how you put things back in to produce something else.
Okay.
No, there's no easy way. You have to know it. You have to memorize it.
Okay. Thank you.
Yep. All right, so briefly for chapter 9, which is probably the longest one out of this set, and for most of you, various presentations, right? So some of you, if you've already had Chem 102, this was like a day in the park. If you've already had, if you had a really strong Bio 1 that spent a lot of time talking about replication, transcription, and translation with all the details, this is probably just a recall for you. If you did not, then this is a lot. Chapter 9 is huge, right? So the first portion about the structure of DNA, which bases, why it's complementary, what a 5' end is, what a 3' end is, again, that should all have been reviewed. For the replication portion, I do expect you to know all of the enzymes and their functions for replication, and they're probably in a purple box in your textbook. And you do need to be able to describe or recognize the steps, the order of the steps, what's different about the 5' to 3' piece being replicated versus the leading strand versus the lagging strand. And then for translation, transcription, same. there's a lot fewer enzymes because it's basically just RNA polymerase, but you should be able to know what the transcription bubble is, the order of events, where the RNA polymerase binds on the DNA. This is necessary and important so that you can understand the lac operon, which comes later, right? And then translation, of course, how we get that messenger RNA to be converted into protein or amino acids. Again, that's why you're going to need the chart. And then this whole other section, the gene regulation section, which I think we did talk about last week, right? We talked about the lac operon. All right, that's related to transcriptional control and how most genes in most organisms are regulated at transcription, meaning if it's transcribed, it's going to be translated. If it's not transcribed, if transcription is blocked, then a particular protein is not going to be made. And because in bacteria, sources of variation, so when we're thinking about evolution in prokaryotes, right? So sources of variation are the same as you learned in Bio 1, except bacteria don't reproduce sexually. So they already have this decrease of variability because they aren't undergoing meiosis. So there's no segregation of chromatids. There's no independent assortment amongst the genes. And there's no recombination, right? So there's no switching of parts of sister chromatids. And there's no, we're not going back to 2N where we have 2 copies of each gene and maybe we'll use 1 copy or maybe we'll use the other copy depending on the environment that a particular organism is in. So how can bacteria evolve so quickly and how come they do evolve so quickly? Well, because they have other methods. Of course, mutation is an important source of variability, any change to the DNA. And in bacteria, because each bacteria only has 1 chromosome, meaning 1 copy of each gene normally, if there's a mutation to that gene, the bacteria is affected. It's either going to be able to do something better or worse, like immediately. So mutation is very important, which is why we go through that. There's a whole section about mutations and how to test for mutations in bacteria, which is the Ames test, right? And how the Ames test relates to if something is carcinogenic, right? And how it's a simple test that can be done with bacteria so that you don't have to spend a lot of money testing mice if you don't need to. And then, of course, the reason I'm bringing up this sources of variation is because of horizontal gene transfer. Aji, can you mute your microphone? Because I'm hearing myself in the background of you. Thanks. So horizontal gene transfer, which is, it's, you know, people say bacterial sex, but it's not really sex because sometimes the bacteria are getting extra copies of things and sometimes they aren't. Sometimes they're getting, sometimes a piece gets cut out that gets lost. But if you want to think of it that way, that's fine. If it helps you remember that it has to do with the variation, that's fine. And then in that particular lecture, I go through all of the different types of horizontal gene transfer. So conjugation, transduction, transformation, and transposons. And yes, you need to know what all of those definitions are and what that means to move DNA in this manner from one cell to another, from one bacterial cell to another, because that's going to come back to us in chapter 12 when we talk about how bacteria develop resistance to antibiotics so quickly. One of the main ways is because they mutate, and the other way is because of this horizontal gene transfer. If I'm a bacteria and I have an antibiotic resistance gene and I'm capable of conjugation, I can give it to anybody I can touch. And because we're bacteria, we're living in a big pile. We're living in a colony where we're all touching lots of other cells, right? So then that mutation can spread very quickly, and a population of bacteria can become resistant to a drug very fast, a couple weeks, less than 2 weeks. So that chapter has a lot in it. right? So it has replication, transcription, translation, gene regulation in the form of lac operon, and then also mutations, which I don't remember if they put after translation. That's usually where they put it before this. And then that whole section on horizontal gene transfer. So all of that is obviously included on the quiz. And then for chapter 6 about viruses, there's a lot less because the chapter is shorter. There is a note outline for that, and I would have, if I were you, I would have watched the video and I would have taken notes in that note outline and expanded it into a full set of notes, because the parts of that chapter that I go over specifically are the parts that are going to be on the quiz, just like every other quiz. What gets put on the quiz is what's in the lecture videos, not what's in the homework. Some of you insist on continuing to email me asking if all the questions from the homework are going to be the quiz. And I keep repeating, no, they're not. No, they're not. And we've gone over this a lot, in my opinion, but you know, I get it. People aren't watching the videos. So you got to watch them and take notes and see what applies to this semester as well as last semester. So chapter 6, that's all of chapter 6. I would say there's probably like 1 or 2 fewer questions from chapter 6, just because it's a shorter chapter. Is there anything in particular that anybody wants to talk about from chapter 9 before I say a few more things about chapter 6? Yes, no? All right. And then, yeah, I think there was just, I don't know what else that I did want to say about chapter 6, except I would say one of the most difficult things for students to keep straight in chapter 6 is that it's really about 2 types of viruses, bacteriophage. Those are the viruses that infect bacteria, and animal viruses. Right? And animal viruses obviously are viruses that infect animal cells, not just human cells. Right? Animal viruses that infect animals. Animals. So I in the video and on the notes, I do the bacteriophage first, because once you understand sort of what we refer to as the multiplication cycle of the bacteriophage, then it's easy to talk about animal viruses, because it's very similar except for a couple of differences. There's just a few differences, but if you already understand the steps involved, and again, that's a process, so you need to know all the steps, you need to know what happens in each of the steps, you need to know the order of the steps, the differences between lytic viruses, lytic phage and lysogenic phage. All right, and those cycles and the difference between lytic and lysogenic, again, are also important because it's very similar to what happens in animal viruses. So some animal viruses also incorporate and become what we refer to as proviruses, just like bacteriophages can become prophages, right? So that's an analogous term, right? All right, other questions?
Dr. Stewart.
Yes.
Just to kind of reiterate that, as far as the lytic phage and the lysogenic phase, just to kind of clarify, for the, I guess I was getting a little confused on the lysogenic. For the second part, it would, it's absorb, penetrate, recombination, and I was getting confused on the last 2 as opposed to the lytic, where it was penetration, duplication. Maybe if you could just go over that second part, the lysogenic, one more time, maybe.
Sure. So here's the virus, or so here's the E. coli, right? Here's the virus. Let's make the virus DNA green. Yeah, that's too close. Let's make it yellow. Right, so in the case of lysogeny or a lysogenic phage, so that would be a lambda phage, is the poster child for that, and its host is E. coli, just like the T even phages are the poster child for lytic phage, and their host is E. coli. So with a lysogenic phage, when the phage, your textbook has super oversimplified this. If you go study genetics or molecular biology, this is really way more complicated. But this is an introductory, right, survey chapter about viruses. So when the virus injects its DNA inside the host, what really happens, and is not described in your textbook, but since I'm on the topic of it. So here's the host chromosome. What really happens in a lysogenic phage is when that DNA comes inside, it forms, but it makes itself look like a plasmid. So it forms a circle. And basically what that does is it helps protect the virus, as it's evolutionary technology, if you want to think of it that way, of the virus. So it protects the virus from being degraded by the enzymes inside the E. coli that normally degrade any single-stranded or double-stranded RNA or DNA that is found in the E. coli cell that's not E. coli DNA or RNA. Right. So if you know a little bit about CRISPR, this is similar to the CRISPR system, right, which was first discovered in a bacteria, because they chop up any single-stranded or double-stranded random pieces of DNA that don't belong to them. And now what we know is they actually incorporate those pieces as well, sort of like as a memory. So the next time that that particular phage tries to infect this particular E. coli cell, it may be able to defend itself. But I digress. So here's the phage that's been circularized, we say, right? And then what happens next is one of two things. There are promoters here on this phage DNA that are either going to, and it's relatively random, they're either going to go to the right or to the left, meaning they're going to bind and start transcribing proteins, either this direction or that direction. If it just transcribes in one of the directions, the phage actually just goes right into a lytic cycle. If it transcribes the opposite direction, what proteins are made are proteins that are going to allow for the recombination event to put phage into this particular E. coli cell. So let's assume that that's happening, because that's what's happening in your book. And I'm going to simplify my chromosome drawing here and not try to draw the double helix portion. I'm just going to put, so there's the E. coli DNA, and the green DNA is the phage DNA, right? So now E. coli lives its life, goes along, right? Multiplies every 20 minutes, remember, because it's E. coli. So after 20 minutes, now there are 2 E. coli that both have phage incorporated into them. And after another 20 minutes, there are going to be 4. And after 8 hours, there are going to be more than 1 million of these. And this is awesome for the phage, because while it's lysogenic, right, it's actually being multiplied, but it doesn't, not having to do any of the work. It's beneficial to the E. coli, remember, for the reasons that are listed. There were 3 on the handout, right? There are benefits for the E. coli to have a lysogenic phage, because many times the phage DNA brings bonus genes into the E. coli chromosome that the E. coli didn't have before, that help it make resistance to antibiotics, help produce toxins, help, and it also helps prevent what we call superinfection. So this may go on for, I don't know, it may go on for a day, may go on for 2 days, right? E. coli multiplying, phage multiplying, until the E. coli, until the host cell experiences some stress. So we'll make the stress red. Help me, I have a test today and I didn't study enough because this is hard. Right, okay, so for bacteria, that it's not usually a test, it's usually temperature, which is why I made it red. It's usually temperature, or more often, how much water is around, right? And what is the pH? Because if the solution or the medium that this bacteria is growing in, if it becomes too acidic, the bacterial enzymes are going to stop working, and obviously it's going to kill the bacteria. So these stressors of temperature, how much water is available, what is the pH? When this E. coli gets stressed due to those factors, that's when the phage, because it's more, again, it's more complex than I'm telling you, but I'll just tell you here. It's because where the phage integrates is a particular spot on the E. coli chromosome, and it's close to the genes that we call the heat shock genes, because some of them have to do with temperature, but others have to do with water and pH. And so when the E. coli actually starts transcribing those genes to protect itself or to save itself, that's going to trigger the phage. That's the clue for the phage to be like, oh, cell's going to die. I need to cut myself out. So the phage DNA is then going to be cut out. And now it is going to enter a lytic cycle. It's going to focus on producing more viral DNA. So I'm going to put more phage DNA and phage proteins. Right, so remember, that's what every virus has to do. It has to produce its genome, be it DNA or RNA, and it has to produce its proteins. And again, remember, it's going to be using the host ribosomes for that. This is an E. coli, so there's no endoplasmic reticulum, right? There's no Golgi, but it is going to use the host, all the amino acids that are here, and these are supposed to be ribosomes that are in the E. coli cell to produce lots and lots of phage, the phage robot army, right? So the E. coli DNA, it's over here, just kind of hanging out, trying to save itself, but it's not working. And the phage, meanwhile, is producing hundreds or at least tens of, there's our little tail pieces, little tail fibers. And well, I made the DNA green anyway, didn't I? So let's put the phage DNA in there, right? Multiple copies of the phage DNA. And then that's going to obviously, sorry, that's going to, I lost my cell. That's going to be able to break open, because the last enzymes that are produced from the lytic phage are going to be enzymes. It's going to be lysozyme that's going to break open the E. coli so that the phage army can escape. There they are. Off they go to infect another E. coli cell. Better or worse, Charles?
No, that's much better. Thank you for the clarification. During
You're welcome.
the actual lecture, there were just certain parts where, like, when you turned away from the camera, it was a little hard to hear you, and I had my headphones turned all the way up, but I was just really trying to get an understanding of it. That's all.
Yes, got it.
Thanks, I appreciate that.
Yeah, relatively low tech here in the Stewart Zoom room. All right, anyone else? Any other questions, worries, concerns? Anything else you want reviewed? So the exam is open till midnight, well, 11:59. Please don't wait until 11 o'clock to sit down to take it, because everything else that's due from every other class, all those students are on Canvas at that same time. So if you have spotty internet, waiting till the last minute is probably the worst thing you can do, right? And just try to get it done before the deadline, and then we'll move on to some new material, chapter 7, a little bit of 3, 11 and 12, which is not as chemical, it's not as biochemical, not as much biochemistry, just a lot more memorizing things. But I think slightly, in some ways, it's a little drier, but it's also a little more interesting because it talks about bacterial growth and how you feed bacteria. And then chapter 12 is all about antimicrobial drugs that kill bacteria, fungus, and viruses. Plus, we also have our microscope labs to do. So that kind of makes it a little more interesting. And then after spring break, we'll start with the growing of the bacteria. And again, just as a reminder, when you start growing the bacteria, you have to tend to them almost, you have to check on them every day. It's worse than having a baby almost. Well, you don't have to do it as many times as a day as take care of a baby, but you do have to check on them every day, preferably about the same time of day. And if they're growing and they've grown a lot, then you have to do something. If they haven't grown enough, then you just have to wait until tomorrow. So you kind of have to have a 2-week, I'd say like a good 12 to 14 days, 9 on the short side, if the bacteria are growing quickly for you, where you can do something for about an hour every day if you have to. Sometimes you can put it off, sometimes you can't. So there's a proposed schedule that I included that I'll put up in that module. I think it's in one of the Carolina modules or in the lab module. And you know, you'll have to take a look at it and kind of figure out how it'll fit into your particular life. But I did just watch some of the video for the microscope itself. There's actually where I go through how to use the microscope. And I did just learn something about microscopes, believe it or not, even though I've been using microscopes for a very long time. Your microscope, your little, so here's the 4X objective, the red knob, right? And I'm trying to get it so that you can see the whole microscope. There we go. Put it back here. The dial on your condenser does change like an iris diaphragm. There's all the way open. There's one that's a little less, and then there's one that's smaller amount of light. That'll help with how much brightness hits your eye and if it's too bright for you to see or not. And then they do also include a red filter and a green filter and a blue filter. It's the blue filter that you want to try to use if possible. Just give it a whirl, because that should help a tiny bit with resolution. So if you think back to chapter 3, which I know for you, you're just like, I don't have time to think about that now. I'm thinking about chapter 9. I get it. But if you think back to chapter 3, where we talked about resolution being the minimum distance that's needed between two objects for you to see them as two separate things, it turns out that blue light is a shorter wavelength than white light. And so you can actually get a tiny bit of increase in the resolution. I think it's 50 nanometers. So it's probably barely discernible through these microscopes. But I think it's worth it for you guys, because you're in microbiology, to at least try to also look at your samples through the blue filter and see if you can see anything more than you could with just looking at them with white light. I don't know if in this particular apparatus that's going to help, but I do know in our full size, my first lab, my second lab microscope, our full size microscopes in Halpern, many of they are equipped with the blue filter. So I would suspect that it does give you a little bit more resolution. The downside is it also is going to give dimmer light. So if you're looking in a microscope like in Halpern at 100x with the 100x objective, and you already are on oil immersion, you would probably have to slide the rheostat all the way up and get the maximum amount of light coming through there before you tried the blue filter. But it's something I think I'm going to try to incorporate into the labs in the fall when we are back in real person. All right. So I am going to end the recording because